Welcome to today's lesson on drugs developed from plants and microorganisms. In this lesson, we will explore the fascinating process of drug discovery and development, specifically focusing on the synthesis of new drugs by chemists that operate in the pharmaceutical industry. We will also examine the interesting fact that the starting point for many drugs are in fact chemicals that have been extracted from plants. In our lesson today, we will explore the origins of these drug compounds that are often found in plants, as well as how they serve as a starting point for drug development and creation. So far, you have learned about antibiotics and painkillers. We discussed how antibiotics can target bacteria to treat infections, as well as how painkillers can alleviate symptoms, though without addressing the underlying cause of the pain itself. This knowledge will help us to better understand the entire process of creating new drugs that can fight diseases and manage symptoms. Understanding how new drugs are synthesized and developed highlights the intricate process of translating natural compounds into effective medications, thus showcasing the collaboration between nature and scientific innovation. This insight is vital for appreciating modern medicine's complexities and the development of future treatments. To understand how modern medicine develops effective drugs, we need to appreciate both historical practices and modern techniques. Many modern drugs have their origins in natural substances, particularly in plants and microorganisms. Plants, in their constant battle against pests and pathogens, produce a variety of chemicals to defend themselves. Some of these chemicals can also be incredibly useful in treating human diseases or alleviating symptoms. Hence, plants can be used to synthesize drugs. Aspirin, which is a widely used painkiller, was originally developed from a chemical that was found in the bark of willow trees. Fascinatingly, the chemical was used in traditional remedies for hundreds of years before it was chemically modified to create aspirin. For example, many Native American tribes in North America were using willow bark for medicinal purposes, long before European colonization, likely for hundreds or even thousands of years.
We gather the bark from the willow tree, knowing its power to heal. When someone in our tribe suffers from a fever, headaches, or swollen joints, we brew a tea from the bark and leaves. The warmth of the tea eases their pain.
Hence, the medicinal use of willow bark is part of an oral tradition that has been passed down through generations, long before written records ever existed. Another example is digitalis, which is used to treat certain heart conditions and was developed from chemicals that were found in the foxglove plant. The plant's active compounds help to strengthen the contraction of the heart muscle.
Upon observing the successful use of the foxglove plant by a local healer in treating dropsy, I became intrigued by its potential. Through careful systematic study of its effects, I discovered that the active component, which I termed digitalis, had profound medicinal benefits. It improved the strength and efficiency of the heart's contractions, providing relief to patients suffering from heart conditions. In 1785, I documented my findings in an account of the foxglove and some of its medical uses, detailing how this plant could be harnessed for the advancement of medical science.
Long before William Withering came to me, we had known the power of foxglove. Passed down through generations, our remedies had healed many, especially those suffering from dropsy, a swelling of the body that you now called oedema. I learned about foxglove from my mother, as she did from hers. Foxglove was part of the knowledge shared by healers like me throughout Europe. In our village and across England, we herbalists had used it for centuries, mixing it into potions to ease heart ailments and other afflictions.
These examples illustrate how compounds that are produced by plants for their own survival can be adapted for human medicine. From microorganisms, drugs such as penicillin can be synthesized. The discovery of penicillin by Alexander Fleming and Myrtle Williams was a landmark in medicine. They observed that a petri dish containing Staphylococcus bacteria had a mold growing on it, the dark pink spots on the diagram, and that the area around this mold was free from bacteria, the light pink area on the diagram. They successfully identified that the mold was secreting a substance, which was killing off the bacteria. They named this substance penicillin, and it became the first true antibiotic. Penicillin has saved countless lives by treating bacterial infections effectively. While many drugs began as natural extracts, today they are mostly synthesized by chemists in laboratories. This shift is driven by the need for large-scale production, consistency, and the ability to modify chemical structures to enhance efficacy or reduce side effects. Drug development often begins with the identification of an active ingredient in a plant, or microorganism. Scientists may screen thousands of natural extracts in order to find a compound with desirable properties. Chemists can then modify the structure of this natural molecule in order to improve its effectiveness, reduce side effects, or enhance its stability. These optimized compounds can then be synthesized in the lab. The newly created substances must undergo rigorous testing. Initial tests on cells and tissues are followed by animal testing. Finally, the most promising compounds enter clinical trials in humans to ensure that they are safe and effective. If a drug successfully passes all stages of testing, it must then be approved by regulatory agencies before it can be marketed. There are many advantages of laboratory synthesis. Synthesizing drugs in the lab allows for precise control over the composition and concentration of the drug. This ensures consistency. Laboratory synthesis can also be scaled up to produce large quantities of the drug. This is essential for meeting the global demand. Moreover, chemists can design substances that are even more effective than their natural counterparts, potentially also with fewer side effects. In summary, many modern drugs have their origins in natural compounds, especially from plants and microorganisms. Examples such as aspirin and penicillin highlight the valuable contributions of natural substances to medicine. Today, most drugs are synthesized in laboratories to ensure consistency, scalability, and the potential for innovation. And the development of drugs involves the initial discovery, structural optimization, rigorous testing, and finally, regulatory approval. In today's lesson, we explored drug discovery and development, focusing on how chemists in the pharmaceutical industry synthesize most new drugs. We discussed the crucial role of chemists in designing and creating new compounds, often inspired by natural sources like plants. Historical examples, such as aspirin from willow bark and digitalis from foxglove, illustrate how nature continues to inspire drug development. We also covered the early stages of drug development, including identifying potential compounds, preclinical testing, and modifying natural compounds to enhance their therapeutic properties. We highlighted the transition from natural extraction to synthetic production, where chemists fine-tune chemical structures for consistency, potency, and large-scale production. Understanding the synergy between natural substances and synthetic chemistry reveals the complex efforts behind developing new drugs and the ongoing evolution of modern medicine.
Watch the rest of the GCSE biology course at Sophos Education.